45 minutes or so. So the title of my presentation is going to be on coronaviruses and disease mechanisms. So I would spend some time explaining the basics of how the virus is confronted in the host system and how the host components are going to react to the presence of the virus in the body. So coronaviruses has hit the global population. So what we know about coronavirus is I think the global population is now aware about coronaviruses. Everyone is now aware of coronaviruses. So there are three basic groups of coronaviruses. So alpha coronaviruses, beta coronaviruses, gamma, and delta coronaviruses. So these coronaviruses are primarily meant to infect apes and mammals. Several species of birds and mammals have found to harbor coronaviruses, where these viruses are meant to live in close harmony with these animal species. So four established coronaviruses have already been reported since 1960. So we have four different coronaviruses which are already there in the human population right from 1960. The first documented evidence arrived in 1960, but I presume that the virus is already there in the human compartment. So you will see that the alpha coronavirus NL63-229E and the beta coronaviruses OC43 and HK1 are already there in the human population, and these four coronaviruses are implicated with upper respiratory infections, and they are associated with causing seasonal cold, and therefore the estimated prevalence of these coronaviruses, these four coronaviruses, has been about 15%, and about 70% of the global population have already become exposed to these four coronaviruses, and we indeed have antibodies against these four coronaviruses. And there has also been some evidence to show that these antibodies and immune responses formed against these four seasonal coronaviruses have implications in reducing or alleviating the severity of COVID-19. And there has recently been a couple of publications providing experimental evidence to show that the immune responses generated against these four coronaviruses have conferred protective attributes against development of severe COVID-19. So, as you are seeing in this slide, there is evidence to show that these coronaviruses have their origins from animal species, especially bats. As you will see here, bats harbor many different types of viruses, many different species of viruses. There have almost been more than 5,000 coronaviruses, found and detected in bats. The microptera and microchiroptera and megachiroptera. These are the two different genera of bat species which harbor these coronaviruses. So of all these different diverse coronaviruses, only about four have been associated with mild disease in humans, and there are three more coronaviruses which I have listed out here: SARS-CoV, MERS-CoV, and of late what we are witnessing, what the globe is witnessing is due to SARS-CoV-2 virus. So all have got their origins from bat species. Okay, so what you are seeing here is the history of the SARS-CoV-2 virus infection, the outbreak that has been reported since December 2019. So there are two important hypotheses that explain that speculate the origin of this SARS-CoV-2 virus, the causative agent of COVID-19. So there are two hypotheses. One is the wet marker hypothesis, and the other one is is the spillover hypothesis. So the disease has got their origin. The disease has got its origin from the Hubei province, where in a beautiful city of Wuhan, you will find the Wuhan University of Wuhan, which is a very popular educational destination also for Indian students. So you will see the virus probably our literature suggests that the virus appears to have originated from the Wuhan wet market, where exotic animal species are normally butchered for consumption. And so it is here where probably the virus might have jumped into the human compartment from exotic animal species, and the disease probably might have expanded beyond the place of origin to afflict the global population. And the second theory, which is the spillover theory, the spillover hypothesis, suggests that the virus probably might have originated from a spillover that occurred at the Wuhan Institute of Virology, where these virus species are normally used for experimentation purposes. But this hypothesis is these two hypotheses, again, I must say that these are merely hypotheses, and there is no concrete evidence to show that the virus originated from Hubei, or the virus probably might have taken its origin from a spillover from the Wuhan Institute of Virology. So there is no solid evidence to show that these two components might have played a role in disseminating the virus across into the globe. And this is the big picture of what we are witnessing at this until this very moment. So MERS-CoV has got its origin in 2012 in Saudi Arabia, which was the hotspot where the virus so the coming to the origin of okay. So the bat species probably might have been the animal reservoirs of this coronavirus. The virus probably might have gotten into civet cats in the case of SARS-CoV virus, and this probably could have been the starting point for the outbreak that occurred in China with the SARS-CoV-1 virus. And 10 years later, we also witnessed the MERS-CoV outbreak happening in Saudi Arabia in 2012, and this probably might have been due to a virus, the coronavirus, the MERS-CoV virus, jumping from the dromedary camels into the human compartment. So you will also see that bat species appears to have been the reservoir host for the virus, which jumped probably into pangolins, the Malayan pangolins in particular, and this probably might have been might have undergone some genetic mutations before it jumped into the human compartment to cause COVID-19. And you might ask me, how come the human humans got proximal to these kind of exotic animal species? You'll know that these animal species, the Malayan pangolins are normally hunted for their scales, because these scales have got medicinal properties, wound healing properties, and many other different psychotic disorders. So the scales have traditionally been used in Chinese medications, and therefore probably the humans might have become exposed to the recombinant virus that probably might have originated from bats, and this probably became the SARS-CoV-2 virus. And we don't know what's in store for us in the years to come with these coronaviruses. So there have been several findings suggesting that the virus spilled over into the human compartment, and they speculate that it probably might have originated from animal workers, healthcare workers, and the studies have also shown that comorbidities and the age over 65 years, uh, appears to pose a risk for the development of severe, disease due to coronaviruses. So this is a picture that I retrieved from online sources from Times Now, which tells how the wet market in China, operate. So you will see also that the evidence also suggests that almost about 500 coronaviruses have been identified in bats, especially in China, and the estimate therefore explains that the coronavirus diversity is reaching almost close to 5,000. And the SARS virus that was reported in 2002 documented 8,098 cases with 774 total deaths, and this was contained thereafter, all due to very small clusters where the infection outbreak had occurred initially, and then it was in 2012 that the MERS-CoV killed almost about 200 and 866 individuals of the 2,521 subjects who reported with symptoms and signs of MERS-CoV infection. And what we are witnessing is the infection is still there, looming around, and although the clusters are not that large with this virus. The SARS-CoV-2 virus is more related to bat coronaviruses. So this is a paper that has been published in Biotechnology in 2020. It's one of the seminal publications, which has recently been. I understand that the paper has been published in Nature Communications. So you will see that the virus, the SARS-CoV-2 virus, has almost about 80% identity with the SARS-CoV virus that was reported in 2002, but it has more resemblance to the bat coronaviruses. So you will see that 96% the SARS-CoV-2 virus has 96% identity with the bat coronavirus. So you will see that the bat coronaviruses are close. What you are seeing here is the virus, having close resemblance. The gene sequences suggesting that the virus has a very close resemblance with bat coronaviruses rather than SARS-CoV virus, which was reported in 2002. But there appears to have been some degree of heterogeneity, because the bat coronaviruses appears to have huge. There is a huge difference that was reported in the sequence that tells that the bat coronaviruses are not strong enough to bind to the ACE2 receptors that are expressed in human systems. So there is some degree of heterogeneity in this part. Otherwise, the bat coronaviruses are more resembling the SARS-CoV-2 virus. So you will see that the RAT-G13. This is the bat coronavirus, which is having 96% homogeneity with the sequences of SARS-CoV-2 virus. And pangolin viruses, of course, have some similarities, but it's not that close as the bat virus. So what's the deal with the bats? So bats, I said the bats are the ones which carry many viruses. So the notable ones are the Hendra virus, the rabies virus, the Marburg, Ebola, MERS, and SARS. So all these viruses have got their origins from bats. So you will see that rabies, the dogs are the ones which are harboring, the rabies virus, and it's from the dog species that the canine animals that humans contract infection. But it's not so. The reservoir host is what is going to be important. So the reservoir hosts are the bat species. So likewise, you have many other different unidentified viruses which have already been reported and documented from bats. So which can also afflict different wide array of animal species. So you will see that all these have got their origins in bats. So why are bats so so much, why infectious agents like viruses of all these viruses have more affliction to bats. So in bats, there is huge level of tolerance to presence of viruses, and this has very well been proven. Experiments conducted in bat cell lines and with these different coronaviruses and many other virus species tells that bats are more tolerant towards presence of viruses. And even if there is an infection with any of these viruses, bats can tolerate these viruses, or else they might present with low grade infections. So micro chiroptera and mega chiroptera are the different species, small bats and larger bats. So they are the ones which are going to harbor all these virus species. So what is the factor? What are the factors that contribute to a poor reflection of poor reactivity from the side of bats? So the bats will have cellular receptors which are more polymorphic. So polymorphism means that their functional domains that contribute to onset of inflammatory cascades are truncated, and therefore even despite the presence of viruses, pathogenic viruses, bat immune components are not reactive, which is not seen in the case of human system and other mammalian systems, which react to the presence of these viruses and which attributes to onset of disease pathogenesis. And moreover, bat species will constitutively express interferon alpha, and they don't. And interferon alpha is meant to, they are classified under type 1 interferons, which are antiviral interferons. So these interferons are meant to, suppress the viral, presence, and therefore these animal species will not develop any symptoms or signs that normally viruses inflict on the bats cells, the cells of the bats. And sometimes you will also see that these bat strains will have constitutively they express interferon stimulatory genes, and there are many other multiple interferons, especially the lambda interferons, type 1 alpha beta interferons, gamma interferons. So these interferons are meant to be antiviral, and therefore viruses are kept in check in these animal species. And you will also see that there are sensors of viral presence, the stress sensors within the intracellular compartment which belong to the innate immune system. You call these as NLRs, nucleotide oligomerization domain. So you call these as NLRP. There is one NLRP3 which is called as an inflammasome. The inflammasome is a component that can sense cell stress, that can sense the presence of foreign antigens, that can sense the presence of stress inducers within the intracellular compartment, and therefore they can respond by producing interferon beta and also interferon 18, IL-1 beta and also IL-18. So these are two cytokines which are meant to be important to necessitate a mechanism called as pyroptosis, and therefore the functions of these NLRP3 has been largely truncated in bat species, and this very well explains why bats are not responding to viral presence. And moreover, these bat species have also been found to harbor NK cells, which have NK cell phenotypes, which have an expanded array of inhibitory receptors, which can very well tolerate the lack of MHC class 1 components, which normally are required for animals to sense whether the sense or differentiate between self and non-self. And moreover, the sting components. Okay, there are DNA, foreign DNA sensing components in bat species, which are called as sting. These sting components can also occur in all mammals. So all mammals will have, the intracellular compartments having the sting, sensors, which can sense the presence of foreign DNA particles. And you will see that these foreign DNA sensing particles, the sting pathway is very much very much remaining vestigial, and therefore this tells that the viruses, the presence of viruses can very well be tolerated in bat species. And there is also evidence to show that experiments conducted using physiology, pathophysiological experiments conducted, to understand, why bat species are very well tolerating viruses, is also because of the fact that the metabolic stress that occurs in these animals during flight can very well be tolerated. So this is again the very important mechanism which tells why bats are very much, they can very well suppress the reactive or adverse immune response, which normally are meant to be important for clearing viral pathogens in the host. So why has there been a failure to contain SARS-CoV-2? SARS-CoV-1 was very well contained. Immediately the outbreak was contained, and we witnessed only very few number of individuals who succumbed to this infection, but not with COVID-19. Why? What was the reason? There have been a list of mechanisms which have been very well explained here. So you will see that SARS-CoV-1, the spillover reservoir was very well known, and they found that civet cats were the reservoirs which were harboring the virus that was responsible for perpetuating SARS-CoV-1 infection, and therefore these civet cats were culled, to break the chain of infection, which was not so in the case, which didn't happen in the case of SARS-CoV-2 virus infection. So the spillover reservoirs were simply speculations. So we speculate that it might have probably arisen from bat species. We speculated that these viral species might have gotten into pangolins and snake species before it jumped into the human compartment. So these were all merely assumptions, and therefore this was the first thing that one should remember, why the SARS-CoV-2, the COVID-19 couldn't be contained at the outset. And the second mechanism is because most of the transmission of SARS-CoV-1 infection that had occurred in China, in Guangdong province of China, was because it had occurred in a hospital setting, and this provided opportunities for the healthcare system to contain the further spread of the infection from the particular cluster. Okay, so this was one reason. But in the case of COVID-19, widespread community transmission was reported, and this explains why we were not so effective in controlling COVID-19. And no transmission occurred 24 to 36 hours after symptoms, and there were a lack of asymptomatic cases with SARS-CoV-1 infection. And even if there were infections, contact tracing was effective with SARS-CoV-1 infection outbreak, which was not in the case of witnessed in the case of COVID-19. So this is also because of the abundant number of asymptomatic or mild infected cases with COVID-19, because almost about 90% of the global population who were exposed to SARS-CoV-2 virus remained asymptomatic. This is again the most important and the widely accepted mechanism which really helped the SARS-CoV-2 virus to spread beyond the threshold. So the actual COVID-19 storyline is thus far is this. So we have almost about 639 million individuals who have already been exposed to the virus. So these are merely estimates, because we don't know, because we don't know the situation of asymptomatic cases, as I had just a while ago explained. So over 66 million individuals have already succumbed to this infection, and over 12.8 billion doses of vaccines have been administered thus far, globally. And this tells why what the current situation is with this COVID-19. So we have already succeeded in containing the, the, the infection. The death rates have really come down, and we expect that the virus would be cleansed very soon, or else the virus, I think, what in my understanding, the virus is going to be there in the human population. So the virus is trying to adapt itself to the human condition. So the virus is actually a spherical virus, as you will see here. The virus is an enveloped virus. So the most important component that is going to be important for the virus to infect susceptible cells are the spike antigens. So you have the spike proteins. So this is the spike protein which is jetting out from the viral envelope. The virus as a matrix or the membrane glycoprotein, which holds the membrane, the envelope of the virus, which has the spike components. And you will also see that the virus has a single-stranded positive sense RNA, which is about 3,000, almost about 30,000 base pairs. So it is one of the largest RNA viruses known in virology. And you will see that the virus will have many other components. Especially the components which are going to be helping the virus nucleoproteins to get get on hold. It holds the nuclear compartment onto the matrix very well, and you will also see that these components are important for for maintaining the architecture of the virus. So the most important thing for the virus to element for the virus to infect target cell is the spike protein. So you will see that the spike protein is the macromolecular structure of the spike protein has been presented here. So you will see that the spike protein has a terminal knob-like domain. So it's actually a trimer. The entire structure is actually a trimer made of 3 monomers, and you will see that the spike protein has a receptor binding domain, which has been shown here. ACE2 binding SB domain is what is been depicted here. So this is the receptor binding domain, which is implicated in binding the virus to the susceptible target cell, which are normally meant to express the ACE2 receptors. And the virus also has these green ones are the ones which are depicting variable regions. The amino acids in these areas are highly variable. This contributes to the mechanisms that allow the virus to escape from the competent immune response. And you will also see the red ones are the conserved regions, which are not going to show any variations. And therefore, this is something that is going to be important from the infection point of view. So the virus infects the ACE2 proteins that are normally expressed almost on all host cells. So almost a vast majority of the host cells will express the ACE2 receptors. And besides the ACE2, there is also a transmembrane protease, serine protease molecule, which is implicated in necessitating the fusion mechanism of the virus with the host cell membrane. So you can see here, this is the host cell membrane, and you will also see the 2 receptors, the ACE2 and the TMPRSS2 receptor, which are implicated in binding the virus with the host compartment. So when the virus binds to the ACE2, there is the spike domain, the S1 domain has to be chopped off for the TMPRSS2 to cleave the transmembrane channel for it to get the virus fused with the plasma membrane of the host cell. So you will see here, this is the picture telling you how the fusion mechanism works. So this what you are seeing here is the host cell membrane, and what you are seeing here is the viral envelope. The viral envelope is giving out the spike protein. So the spike protein has S1 and S2 domains. So S2 domain is a stalk bearing a stalk-like structure. So you can see here these 3 components are mere monomers, and these monomers are the ones which are going to hold the stalk-like domains, the bulb-like domains, which has been represented here as S1. So what happens here is immediately after binding with the ACE2 protein, the TMPRSS2 will have a furin component which will cleave these two domains in the spike protein, and what is left over is that the S1 component is going to be released, and then you will see that the S2 component is going to form a hairpin loop like structure which holds the virus particle firmly with the host cell membrane, and thereafter the virus is able to come closer to the cell membrane after forming a hairpin loop, and you will see that the virus will fuse with the host cell membrane for it to inject its nucleocapsid into the cell, into the host cell. So ACE2 is normally a protein that is expressed on pulmonary epithelium and the vascular endothelium, and ACE2 is meant to lower the. So this ACE2 is different from the ACE2 which normally we see in the respiratory as an ectoenzyme expressed on the apical surface of the pulmonary endothelium. So that ACE2, the other ACE2 other ACE is different. That ACE is different. Angiotensin converting enzyme is different, which is meant for maintaining the blood pressure, increasing the blood pressure, which operates through the renin-angiotensin aldosterone system axis. This ACE2 is different. So this ACE2 is totally different. So you will see that the ACE2, this ACE2 is meant for lowering the blood pressure by cleaving ANG1 and angiotensin 1 and angiotensin 2, which is having an opposing effect as that of the ACE component that is found on pulmonary endothelial cells. So the ACE inhibitors do not affect the ACE2, and therefore they may contribute to lung pathology and bronchoconstriction. So I will explain all these details in the pathophysiological mechanism of COVID-19. So this is the genomic structure of the SARS-CoV viruses, SARS viruses. So you will see that all these viruses will have 1A or open reading frame 1A, 1B, and then you will also see that all these three components are going to be, all these components, the 1A and 1B or 1A and 1B are almost similar in all the coronaviruses, and you will also see that the spike regions are also almost the same, and the architectural gene architecture across all these coronaviruses are all the same, but there are some minor differences which you will see occurring in the spike proteins, and this contributes to the heterogeneity of these virus species. And you will see these proteins will have the proteins that are translated from these primary transcripts. mRNA transcripts are meant to have different structural, as well as they play a role in structural as well as non-structural functionalities of the virus. So this picture tells you how the virus is going to infect susceptible cell. So the virus is binding with the ACE2 and the TMPRSS2, and then the virus is getting endocytosed, and then the virus gives out the RNA. The RNA genome is getting injected into the cytoplasm. Being an RNA virus, the replication occurs in the cytoplasm. The RNA is of positive polarity. So the virus is able to synthesize, make use of this positive polar RNA as an mRNA. It can translate proteins. So the proteins that are formed initially are the polyproteins. So these polyproteins have to be broken down by proteinases that the virus hijacks from the cytoplasm of the host cell, and then the virus will also make use of the RNA-dependent RNA polymerases from the host compartment, to synthesize more copies of RNA, genomic RNA, as well as mRNAs for it to be translated into viral proteins. So you will also see the classical mechanism of protein synthesis occurring before the virus is packed and exocytosed from the cell that the virus has infected. So the virus is a cytopathic virus, so it can kill the cell that it infects. So virus transmission occurs through the respiratory tract predominantly, although there have been reports of gastrointestinal COVID and many other mechanisms whereby the coronaviruses can enter into the host system. So these coronaviruses, whatever I am explaining, is a generalized component. The pathophysiological mechanism is all the same across all the coronaviruses, be it the SARS, be it the MERS-CoV or the COVID-19 causing SARS-CoV-2 viruses. So you will see that the virus infects predominantly through the respiratory tract. The upper respiratory tract has the epithelial cells. The airway epithelial cells will express ACE2, and therefore they may become infected with the SARS-CoV-2 virus. The SARS-CoV-2 virus is the example that I have given here. So you will see that the olfactory cells are also getting infected. Olfactory epithelial cells will express enormous amounts of ACE2, and therefore they become susceptible to infection by SARS-CoV-2 virus. So you will see that of these different cells in the olfactory compartment, the multiciliated cells will express high levels of ACE2, and you will also see that some of these the other cells, the basal cells especially, and also the suprabasal cells, also to a lesser extent will express these ACE2 receptors. But again, the mucociliary escalator also is responsible for clearing the virus species, viral particles from binding to the cognate receptors. So when there is individuals, when individuals smoke, or when individuals have an issue with the mucociliary escalator, the chances for the virus to collide with the receptor, the ACE2 receptor, becomes much easier, and therefore individuals with a poorly functional mucociliary escalator component are more vulnerable to become infected with SARS-CoV-2 or the other coronaviruses. So after entering into the respiratory tract, the virus the virus gets down into the lung compartment. You will see that the viral components fusing with the alveolar walls. So you will see the alveolar structure like what you are seeing here. The alveolar compartment is in close contact with pulmonary capillaries. The alveolar wall is actually made of two types of cells, one the type 1 pneumocyte and the type 2 pneumocyte. Of these two pneumocytes, you will see that the type 2 pneumocytes will express enormous amounts of ACE2 protein. So this is evident from research which was recently published in a journal called the Pathology. There is a pathology journal which tells that the quantum of expression of ACE2 proteins on the type 2 pneumocytes is too humongous, and therefore these cells are going to become readily infected, going to receive the virus readily, more than even the type 1 pneumocyte. So besides these two cells, you will also find alveolar macrophages, also called as dust cells, in the which patrol the alveolar compartment for presence of any virus particles, and therefore these cells are also going to play a very important role in disseminating the pathogenesis. So I'm sure the audience is aware that the type 1 pneumocytes are going to be important for facilitating gas diffusion across the respiratory membrane, and type 2 pneumocytes are important because they produce surfactants which are responsible for providing facilities for oxygen to move across the respiratory membrane. So you will see that the respiratory membrane is a very delicate structure which is made of in the endothelial cell, and then there is also a fused basement membrane, and towards the other side you will also see the alveolar epithelial cell, and these alveolar epithelial cells would be covered with surfactants for facilitating a gas exchange. And this membrane, the thickness of this membrane is just 0.5 microns, and any damage that is inflicted, that the respiratory membrane inflicts, is going to affect the going to lead to respiratory pathologies. So what you are seeing here is that the virus is getting into the respiratory tract. So after getting into the respiratory tract, you will see that the virus is going to infect the type 2 pneumocytes. So once the type 2 pneumocytes become infected, what's happening is that the virus causes injury, mechanical injury to the cell, and now this alerts the infected type 2 pneumocyte to release danger associated molecular patterns. A bunch of danger associated molecular patterns have been reported in COVID-19. HMGB, you have IL-1 alpha, and you have also IL-33, and there is also heat shock proteins. So these are the alarmins, are the danger associated molecular patterns that these infected cells release at the outset after getting exposed to the SARS-CoV-2 virus. And you will see that the cells that are patrolling the area are the macrophages. The macrophages arrive at this site because they are already there in this area, and you will see that these macrophages attempt to help the cell, but these cells become activated and they start to release endogenous pyrogens, which are the IL-1 beta, IL-6, and TNF alpha, which are also called as the endogenous endotoxins. So these components are going to be important for inducing acute phase responses. You will see that these three components can readily diffuse into the blood circulation, and they can move all the way to the brain. They can get into the anterior into the hypothalamus, and they can tune the thermoregulatory center to induce fever. As a result of this, the hypothalamus will produce prostaglandins, leukotrienes, which are mediators of fever and inflammation. And you will also see that the individual will start to experience rise in body temperature. And the next thing is that these components can get into the liver, and they can direct the liver to release acute phase reactants, especially complement, ceruloplasmin, and IL-6, and many other components. They can also go to the adrenal gland. They can also direct the adrenal gland to release adrenaline, and therefore they can also be held responsible for the release of corticosteroids, which can also be helpful in necessitating inflammation. And what happens also is that the blood that is getting pumped into the pulmonary capillary due to this injury is going to bring lots of inflammatory cells and also many other inflammatory components. One of those inflammatory components is IL-8 that is getting released by the macrophages. So these IL-8, also called as CXCL8, is going to recruit neutrophils. So neutrophils are the first cells to arrive at this site of inflammation, and you will see that these neutrophils are the ones which are responsible for necessitating or magnifying the pathogenesis mechanisms that we see normally in coronavirus, especially the deadly coronaviruses, the SARS-CoV-2, SARS-CoV, and the MERS-CoV infections. So you will see that the virus is getting here, and it is going to create all chaos. So the blood is now here. The blood is going to the blood cells are the blood vessels are going to dilate, and this leads to escape of fluid, plasma, into the interstitial compartment. There is plasma leakage. There is a vascular permeability, which is going to be appreciated as one of the critical signs, one of the key signs of inflammation: heat, redness, pain, and swelling. So you will also see that eventually the blood becomes much thicker. The blood also lacks oxygen, all because of the excessive accumulation of fluid in the alveolar lumen. And you will also see that the respiratory membrane gradually becomes thicker due to deposition of metalloproteins and also collagens and so on. and as a result, what happens is there is, poor diffusion of oxygen from the alveolar lumen into the pulmonary capillary. So as a result of which, we appreciate a drop in partial pressure of oxygen, and this partial pressure of fall in partial pressure of oxygen is what is called as hypoxia. And you will also see that this poor perfusion of oxygen in the blood is going to be sensed by the peripheral chemoreceptors. So the peripheral chemoreceptors are located in the aortic arch and the carotid arch. So these are going to be impulses are going to be generated, and these impulses are going to be relayed through the glossopharyngeal and the vagus nerve, the cranial nerve number 9 and cranial nerve number 10, and this is going to be relayed to the respiratory centers in the brain stem, the dorsal and the ventral respiratory groups, the apneustic and the and the and the the other respiratory centers, the pneumotaxic respiratory centers, and you will see that the impulses are going to be brought back to the rib cage, and as a result of which the individual will have to exert more pressure for inspiring, and this is something that is going to be responsible for breathlessness. So these are signs which tells that the level of oxygen in the blood is less, and this is what is going to increase the work of breathing. Okay, and then the chemoreception is also going to be sensed by the sympathetic nervous system, and you will see that the individual will have difficulties in breathing, and there is also pulmonary edema and many other pathogenesis mechanisms. And you will also see that the blood has become more thicker, and you will see that the perfusion of blood to different blood getting into the different organ systems are going to be affected. You will see that the perfusion is getting affected, and because of this, there is influence of multi-organ failure. The poor perfusion, poor blood volume, and poor blood pressure leads to cardiorespiratory arrest. So this is what is going to be the pathophysiological mechanism underlying the onset of severe COVID. So this is the big picture of what exactly is happening, during the pathogenesis of SARS-CoV-2 infection. So I think if Dr. Sri can permit, maybe one more slide.
Yes, sir, please.
Okay, because I could hear some background.
No. Participants joined, and it's okay, sir. You can.
Okay, maybe I will go with this one slide, and then
yes,
maybe
we can have some interaction.
And we can continue the presentation maybe next week or so.
Yes, sir.
So what we see here is the difference between children and adults with the disease presentation. So the classical mechanism, whatever I had explained, is appreciated in adults, and in children what we see is that the expression levels of ACE2 is very minimal, and this attributes to poor pathogenesis mechanisms, poor influence of pathogenesis mechanism, and also because these children normally will retain the Th2 profile, because it takes a while for them to switch over to Th1, and this explains why Th2 is more towards the suppressive side. It can produce cytokines that can suppress the immune system, and moreover Th2 individuals will also tend to have more of regulatory T cells. These regulatory T cells can also suppress the untoward adverse immune responses, and therefore this explains why children normally will not have a pronounced severe COVID infection. So I think with this mechanism, I will stop my presentation.
Yes.
I will continue my presentation explaining the other pathogenesis mechanisms